Some years ago, the conventional wisdom was that the brain developed. When it developed, it formed its connections. And once it was developed, it remained pretty much stable throughout most of adulthood until people began to age, and then maybe some of the connections were lost and maybe some of the nerve cells were actually lost. What we've found more recently, by examining animals that grow up or are as adults placed in these enriched laboratory environments, is that the brain of the developing and the adult animal continues to be a very dynamic place. New synapses are forming, new blood vessels are forming. In general, the organization of the adult brain continues to reflect the history of that brain. It's as if in preparing for the future, the best way to do that is to adjust on the basis of what's happened in the past. Several years ago, we did experiments in which we housed animals in two different environments. One, which we called the enriched laboratory environment, had a group of rats in a large cage filled with toys, play objects, exploration objects that allowed for learning. The objects were changed every day so that the animals experienced a new environment each day. The contrast group, or control group, if you will, was a group of animals housed in a standard laboratory cage, just a clear plastic cage that allowed them to see out into the room. They were housed alone, no other rats in the cage. What we found with those two environments is that the animals in the complex or enriched environment developed nerve cells in various regions of cerebral cortex and other parts of the brain that had larger dendritic fields. That's the input side of the neuron. And those dendritic fields actually had more inputs, more connections from other nerve cells, so that the wiring diagram was actually embellished in those animals relative to the animals that lived in relatively impoverished cages. Another thing that we found is that the vascular supply, the blood system, was actually embellished in these animals. The animals had more capillaries. To really understand how important that is, you need to know that the brain more or less runs on empty. It can't store energy, carbohydrate or fat, in the way other parts of the body do. And so it's critically dependent, moment to moment, on the blood supply for its nutrients. And hence, if we tax the brain more, it adjusts in ways that allow it to respond to the peak level of demand it's likely to experience. When we were doing the work with the enriched environments, we thought that everything sort of happened together, that it was one single orchestrated process. Nerve cells changed, blood vessels changed, and actually something I haven't spoken about, supportive glial cells all changed together. In order to examine this more closely, we did experiments where we separated out exercise from learning, so that we had one group of animals that learned motor skills where essentially they had to traverse an elevated obstacle course. These animals were learning, but they weren't getting a lot of exercise in the course of that experiment. By contrast, we had animals in exercise wheels that got a great deal of exercise, but had relatively little opportunity to learn. The somewhat surprising at the time result is that the animals that had the opportunity to exercise, and I should say these were adult animals, these were not developing animals, the animals that had the opportunity to exercise had a selective increase in the density of vasculature and no real change in number of connections. The animals that learned the obstacle course had a change in the number of connections, an increase in the number of connections. Again, the same general range that we talk about with the complex environment, 20 to 25%, and no change in the density of blood vessels. So what this shows is these are independent processes, and different demands placed on the brain will cause the brain either to change its wiring diagram to incorporate learning and memory, or change its blood supply to allow greater stamina to drive the muscles of the body. With regard to the implications for adult humans, we haven't really done those experiments yet. There are a few experiments that suggest that exercise can improve cognitive ability in sedentary humans, and that presumably is working through these kinds of mechanisms. But the one thing we can say is that the brain of a rat is very similar at the level of the individual nerve cells to the brain of a human. And what we see in rats, we really would expect to see equivalent effects in humans.